our model of, and I'm going to comment on that too, but I just want to just make it clear to everyone listening. Like, if we look at our model of pre-existing heterozygosity, would you say that science, you need to have testable and falsifiable predictions, right?
Sure, and
Because,
that can
okay,
be. I mean, that can be done fairly easily, right? I mean,
I just want to let you, because obviously, we've talked in detail about, the non-protein coding regions, pre-existing heterozygosity, but we're saying that obviously this, most of the non-coding DNA will be functional, okay? And obviously we would probably agree to disagree that what we're seeing is functional. I would say that it's clearly not idle, but here's the big thing, right? Differences in, say, mitochondrial and nuclear DNA sequences between families are expected to reflect functional differences, but not just due to neutral mutation. Okay, because if we believe in original created kinds, we're going to agree in function. Okay, genome-wide function, but different functions across families. Because we would say that the created kind is, you could probably take it up to to the family level, but measured mitochondrial DNA mutation rates and known mitochondrial DNA differences, say within families, they would be expected to fit the creation timescale and what we would assume is a constant mutation rate, because the created kinds, and obviously if you understand the model, they're obviously going to start with no mitochondrial DNA mutations, right? Because we inherit mitochondrial DNA through what? Our mother, right?
okay. I mean, I think even if you're going by creationist standard, you would still assume that I wouldn't grant you that there's a constant mutation rate because, and again, we seem to both agree to the fact that the environment can cause or can dictate the rate at which mutation can be affected.
But would you
So,
say it's fair that if evolutionists are allowed to inherit mutations, say as the sort of the source of all variety, why can't we as creationists inherit pre-existing differences? Like, would you say that that's fair if we're comparing models?
well, I think you can, but I think that generates more harm than good for you. Because if you do genomic studies across species, you'll find that a lot of species, I mean, humans and apes, and there's quite a few, like mammals, for instance. We share quite a few different, we share quite a lot of genes, and some of them we don't even use. Some of them we really don't have a function for, but they're there because we call it evolution. You guys would call it, I don't know.
Okay, so
But the point being is that we share genes that aren't even within our own family sometimes.
so
So
are
it's
you saying that the environment determining factor is natural selection? Would you be able to make a prediction based on natural selection? Because if we look at these constant mutation rates in regards to mitochondrial DNA, the differences would actually point more towards a 6,000 year timescale versus a 200,000 year timescale, say back to the origin of humans. Go ahead.
right. So I mean, like if I'm going to argue that like temperature, for instance, or the environment can play a crucial role within the mutation rates, I can subject mice to all sorts of different temperatures. I can have some colony of mice that starts off, give them a very happy life, food and water and all the things that a mice needs, and after a certain amount of time I segregate them and I say, okay, so one of these mice are going to live in normal conditions that mice normally would operate under, but some of these mice are going to operate under conditions that would resemble more of a drought situation. So they get less water at certain increments. There's more heat that they're exposed to. They have significantly less rest time. I can create parameters to test, what are factors that could increase mutations within these mice.
Okay,
And
so
at the
can you fill in the blanks, say, of how much natural selection then has to eliminate in order to, say, fit within the evolutionary theory? Like, would you be able to sufficiently fill in those blanks and then make that successful prediction?
I'm not sure if eliminate by you mean by meaning like silencing of certain genes, or if you mean just out and out eliminate, the gene itself.
Well, I'm just saying that it seems like there's a lot of guesses, which means no testable prediction, which would indicate that it's obviously not scientific. Because from my understanding, most nuclear DNA differences, if we're looking at say equids, between them, they're homozygous within those species. So if we look at the 26 million nuclear DNA differences, let's say between the imperial zebra and the domestic horse, just 2 million are heterozygous within the domestic horse. And we actually see that other species display this same pattern. So if we actually look at all the nuclear DNA differences, they're significantly more than obviously the mitochondrial DNA differences. And we would say that's because of pre-existing nuclear DNA differences. And we would say that the mitochondrial DNA differences are based on a constant rate since Eve, right? Because we pass it down from Eve. Go ahead.
Well, I guess to some extent that would hold. I mean, the first part of what you said I don't agree with because of the fact that, sure, it's hard, but that's also why people are invested in the field of science. I mean,
well, how much? Okay, well then I guess it's simple. Then how much mutations does natural selection have to eliminate then? Like obviously, for it to be testable and falsifiable, and you need to fill in the blanks, and you need to make a prediction on natural selection. How much mutations does natural selection ultimately have to eliminate? Go ahead.
I mean, you're asking. Okay, so in a controlled experiment, we may be able to guess that.
Are you currently testing it, or are you saying that this is the prediction?
So what I'm saying is that, like, if I were to set up an experiment with mice under conditions that I knew, I could probably make reasonable predictions about how many mutations occur per generation or something to that effect. But for me to say, here's how many mutations needed to occur for, this mice to go to something that's, a different species or something like that. Okay, well, I would need sufficiently more data about the environment, about how these mice were living, what kind of pressures are they under, things like that. I wouldn't have enough information to judge that.
Okay, so what you just said is you can't, and it's only a guess. Now, let's look at the family of obviously cattle, sheep, antelope, because this is one that we can actually look at, okay? Because they have been analyzed. So there are hundreds of species in this family for which there is ample nuclear DNA and mitochondrial DNA information for analysis, okay? Now, if we look at it, and I urge you to do so, there are only a few thousand mitochondrial differences separating any two species within this family, which is amazing because what we do then is we draw a branching tree from these data, okay? Now, what the tree shows, it shows splitting, okay? So the distances between the splitting events, let's just say the branch points to new species, are reflective, obviously, of the number of mitochondrial DNA differences. Now, what I said earlier, we're assuming a constant mitochondrial DNA mutation rate, okay? Now, the plot shows the number of species as a function of time. So what the plot suggests, if you're following me, is that there is a constant mitochondrial DNA mutation rate within families, and the mitochondrial DNA mutation rate per species decreases as homozygosity increases. But for nuclear DNA, and I'm almost done, speciation decreases heterozygosity and hence the potential for speciation. So if we actually look at the species that have been analyzed, it's more consistent with our model of pre-existing DNA differences and constant mitochondrial DNA rates. Go ahead.
Well, sure. I mean, that's all well and good. The first question, of course, is, you're dealing with you're dealing with some amount of species. That's fine. Certain species of animals have different means of survival mechanisms. Some are engaged by sex arrangement. Some are engaged by hyperactivation of certain chromosomes. Of course, the thing that has to be asked is this constant just throughout this particular selection of species that were chosen, or is this constant throughout all species?
Well, obviously, Well, I mean, we've only sequenced certain species. I'm looking at species.
Well, sure, sure. I mean,
so,
but yeah,
well,
I understand that. But of course, though, that's fine for the sample size that they have, but it's still speculative.
And that's our prediction, right? Because my whole point is mutation rates of a handful of species. The whole point of everything I just said is they are consistent. That's how creationists, but not evolutionists, can make predictions on all animals' mutation rates. So whether it's the pre-existing nuclear DNA differences that we say are based on pre-existing heterozygosity or constant mitochondrial mutation rates, we're making the predictions. And so far, the trajectory is pointing towards a stronger model based on a biblical based model. And I understand that we'll see what the future has in store, but some of these testable predictions are coming true, which I'm actually posting in the description box. But it seems like the trajectory is stronger for us than it is for evolution itself. Go ahead.
And I think a few evolutionists have suggested the same thing, that mitochondrial DNA differences may be a contributing factor towards speciation. The question, of course, is because it's young, how much of a bearing does it have on in the rate of speciation? I mean, it could be significant, but it could also contribute 1%, which in that case, that's fine, but that means there's something still larger at play.
But if the DNA differences, say, of all species so far, obviously we haven't sequenced them all, if they're too few based on everything I'm saying, evolutionists have always predicted far too many differences. So why is that the case? And after you explain to me why that's the case, would the rescue device be natural selection? And if it is natural selection, I want a testable, falsifiable prediction as to how many mutations natural selection has to eliminate. And if you can't give me a testable, falsifiable prediction, then it's not scientific. Go ahead. Because it sounds like we're agreeing that based on these mitochondrial DNA differences and nuclear DNA differences, the creation model is the one making the testable, falsifiable predictions, and the trajectory of the non-coding protein regions is pointing towards genome-wide functionality. Now, obviously, we'll see what the future has in store, but what I'm saying is, based on all this current evidence, the creation model appears to be more scientific. Go ahead.